Hi guys, welcome to Supplement Tutorials. Today we'll be talking about the cell membrane or the plasma membrane. In our previous video, we went through all the various topics under cell biology. And today we'll be beginning with the structure of the cell membrane. So the diagram you see here is the structure of the cell membrane. The cell membrane has different components. We have the peripheral proteins, we have the cholesterol, we have the glycoproteins or the glycocalyx, we have the phospholipid bilayer, then we have the transmembrane protein. So when we talk about the cell membrane, for instance, let's consider this to be our cell. So the cell membrane acts as a barrier between the intracellular compartment and the extracellular compartment, or between the inner environment of the cell and the outer environment of the cell. So this is the inner environment, this is the outer environment. So that is the main function of the plasma membrane. It serves as a barrier between the intracellular compartment, which is the fluid or the compartment within the cell, and the extracellular compartment. So that is the main function of the plasma protein. Now, the plasma protein has been equipped with certain components that facilitates the transfer of substances in and out of these two spaces. That is the intracellular space and the extracellular space. And not only does it ensure transport, it's also involved in myriad of functions such as signal transduction, cell communication, cell rigidity, etc. So we are going to dive deeper into the various functions of the plasma membrane and the various parts too as well. So the plasma membrane basically consists of components such as the phospholipids, then we have proteins, then we have the glycocalyx, then we have the cholesterol. Okay, so I think these are the basic components of the plasma membrane. So when we talk about the phospholipids, we have different types of phospholipids. The proteins, we have different types of proteins, the glycocalyx too as well. And then the cholesterol too plays some role in the plasma membrane. So now let's talk about the phospholipid. So normally the plasma membrane consists of a phospholipid bilayer, so that we can see that this is the structure of the phospholipid per the diagram. So it's a phospholipid bilayer. So there are two layers that are made up of phospholipids, and every phospholipid is made up of a tail and a head. So we always say that the head is a water-lichen part, which is also known as hydrophilic. So we have a hydrophilic head and then a water, let's make it hating tail, which is also known as hydrophobic tail. So if you look at the structure of the cell membrane, you realize that the hydrophobic part of the phospholipids are facing towards the aqueous regions. So here, let's consider this side to be the outer side of the cell, and this is the inner side of the cell. Both sides contains fluid, and since the head is water-lichen, it is therefore designed to face either the outer or the inner, the place that contains fluid, or the aqueous place. So that is what makes the the phospholipid bilayer having both hydrophilic head and hydrophobic hot tail. So this is a characteristic that we have to notice. So the head is the water liking part and the tail is the water hating part. So basically there are types of phospholipids that we are going to dive through. So when we take the plasma membrane, these are the types of phospholipids we are likely to encounter. So the first phospholipids, so here let's make here inner. So this is the inner membrane. So the cell membrane, we have the inner membrane and then the outer membrane. So in the inner membrane, these are the phospholipids that constitutes it. We have the PE, which is also known as phosphatidylethylamine ethanolamine or ethanolamine or ethanolamine, however you pronounce it. So this is one of the components of the inner membrane. That's phosphatidylethylamine. So it has an ethanolamine head. It has an ethanolamine head. Then we have the second one. We have the phosphatidylserine. We also have the phosphatidylserine. And then the last one, we have the phosphatidylinositol. We have phosphatidylinositol. So these are the phospholipid components that are found within the inner membrane. These are the phospholipid components that are found in the membrane. So either we have the phosphatidylethanolamine or the phosphatidylserine or the phosphatidylinositol. Then in the outer skirts or in the outer membrane, we have the PC, and then we have the SM. So the PC is the phosphatidylcholine. Phosphatidylcholine. Then the SM, we have the sphingomyelin. Okay, so all these phospholipid components have the basic functions they play. So for instance, phosphatidylethanolamine is responsible for maintaining cell curvature and also infusion process. So here, maintaining cell curvature and fusion process. So when we talk about, let's say, endocytosis, it is this component of the phosphor or the plasma membrane that is key or critical in the endocytotic process or endocytosis process. Then we can also talk about cell curvature, maintaining cell curvature. So maintaining cell curvature, okay. So for instance, when we talk about a pathological condition such as the sickle cell diseases, we realize that there is a twat in the cell's curvature, and we can, per this knowledge, we can say that there might be an effect in this component of the phospholipids that is trying to give that abnormality in the cell curvature of the red blood cell, leading to what we call sickle cell anemia. So this phosphatidylethanolamine, that is the function. Now when we talk about phosphatidylserine, it also deals in cell signaling. So in our future videos, we are going to talk about cell signaling and cell signal transduction as well. So this one too involves in cell signaling, and phosphatidylinositol too involves in cell signaling. Then when we talk about the outer skirt, the phosphatidylcholine is also responsible for the fluidity. So fluidity and then curvature. Then the sphingomyelin too is also responsible for membrane rigidity. So here membrane rigidity. So which is why it's mostly found at the outer side. So they just serve as a barrier and also maintain the cell hot rigidity or membrane rigidity. So these are the major phospholipids that we find. So these are the major phospholipids. That we have. So our next component that we will talk about is the proteins. So embedded in the cell membrane is a protein we call integral protein. So integral protein, what we have to notice is that they are embedded within the plasma membrane. So there are different types of integral proteins that are embedded within the cell membrane. So first of all, we can talk about the channel proteins. We can also talk about carrier proteins. We can also talk about channel protein, carrier protein. We can talk about receptor proteins. Then we can talk about cell adhesion molecules. Cell adhesion molecules, and maybe some enzymatic proteins. So mostly these proteins are found embedded within the plasma membrane. So for instance, the channel proteins, we can talk about the sodium potassium pump or the sodium potassium channel that is responsible for the ion exchange, or responsible for ion balance between the intracellular compartment and the extracellular compartment. So we can talk about the proteins channel too. Then we can also talk about carrier proteins. Carrier proteins are also responsible for the transport of substances. They are substances that are able to move from the extracellular compartment to the intracellular compartment through these carrier channels or carrier proteins. Then we have receptor protein. So there are certain transports that requires receptor. So, for example, we have a process we call receptor mediated endocytosis. These receptor proteins are also responsible for that. And for instance, when there's substances such as insulin, when insulin wants to enter into the intracellular compartment to be used by the cell, it has to find an insulin receptor, and it's a receptor protein, so that binding can induce the uptake of the insulin. We also have cell adhesion molecules as well, and enzymatic protein. All these are integral proteins that facilitate the transport or the movement of substances in and out of the cell, also ensure cell communication and signal transduction, etc. So these are the basic integral proteins that we have. Now, what we have to notice is that the integral proteins are embedded within the plasma membrane, just like these ones. So this one, for instance, are integral proteins. This is an integral protein. It is found within. You can see that there is an embedment of the protein. The protein runs through the plasma membrane. All right. Then, when we talk about the peripheral proteins are often found on the surface of the plasma membrane. So the peripheral proteins, they are attached to the surface. So what we have to notice is that peripheral proteins are attached to the surface. Peripheral proteins are attached to the surface. And then examples, we can talk about the cytoskeletal proteins. So cytoskeletal proteins. So these proteins, we have examples to be the spectrin, we have the actin, then we have the ankyrin. We also have the protein bands. So mostly when we are dealing with the structure of the cell membrane, one of the cells that is most prevalent, is most used, or is most significant in analyzing the structure of the cell membrane is the red blood cells. You know, the red blood cells, they are enucleated. and then we can see that they have a large cell membrane. So when we study the red blood cells, this spectrin, actin, ankyrin, and protein bands, we have protein band 4.1, etc. They are mostly found on the surface of the red blood cells. So they are what? They are peripheral proteins, then they are mostly found on the surface of the red blood cells. And they are also involved in myriad of functions like cell signaling, etc. So aside the cytoskeletal proteins, we also have signaling proteins such as the gene proteins. So signaling proteins. So we have the gene proteins, etc. So all these are what? Peripheral proteins. And in future videos, when we are talking about cell transport and cell transaction, we are going to dive much deeper into how these cytoskeletal proteins and signaling proteins play a role in their various functions. Then we also have lipid embedded proteins. So these proteins are mostly incorporated with what? Lipids. We will talk much about this one in future videos. The lipid anchored proteins is also an alternative form of what? Proteins that are found within what? The cell. Okay. So let's talk about the glycocalyx. So the glycocalyx actually is the surface layer of the plasma membrane. So it's the surface layer of the plasma membrane, and it consists of glycoprotein. So we talk about glycoproteins. Let's break the word down. Glyco. So let's say carbohydrates, hydrates. Then we have protein with protein moiety, protein. Then here we can say glycolipids. We can talk about carbohydrates, and then lipids. Lipid with lipid what? Moiety, lipids. Then we have the proteoglycans. The polysaccharides are mostly what? Sugars. Then we have what? Proteoglycans too as well. So these constitute what's the glycocalyx. One special thing about the glycocalyx is that one, it gives the cell its antigenic what? Property. So when we dive into immunology, for the immune system to recognize a cell, it is based on the glycocalyx. There are certain glycoproteins and glycolipids that gives the specific cell its antigenic property, or allows the immune system to recognize the cell. For instance, when we talk about the red blood, the blood, various types of blood group. So for instance, we have specific glycocalyx here that allows the immune system to identify a specific blood group as type A. We have some that identify to be type A, B. We have some that identify the cell to be type B, and then if it doesn't have that particular glycocalyx, it is named as O. So the glycocalyx provides the cell with its what? Antigenic property. So for cell recognition, it is the glycocalyx that plays a major role in cell recognition, so that the immune system will be able to distinguish between self from non-self. So that is actually the function of the glycocalyx. Now, pretty quick, before we talk about the fluid mosaic model, let's do a brief discussion about the general functions of the plasma membrane. So general function of the plasma membrane, we can talk about transport, and what? Permeability. Then we can talk about signal transduction. Then we can talk about antigenic recognition. antigenic recognition. Then we can talk about cell to cell interaction. Then we talk about, okay, so cell transaction. Okay, we can talk about cell rigidity, cell fluidity, fluidity and rigidity. Then we can talk about the cell protection. So the glycocalyx is also one thing about the glycocalyx is that they're also responsible for cell protection. So the glycocalyx play much key role in what, in cell what protection. All right. So we can talk about cell protection as well. So basically, these are the major functions of the plasma membrane. There are a lot of functions, but I think at least these are the basic function of the plasma membrane. Okay. Now, the final thing we'll talk about is the fluid mosaic model. The fluid mosaic model. So the fluid mosaic model was actually proposed by a scientist called S.J. Singer. So S.J. Singer proposed this model in 1972, and this model is simple. So we describe the plasma membrane as a dynamic and flexible structure that consists of components, as we've already described, like glycoproteins, integral proteins, phospholipids, transmembrane proteins, etc., and then the cholesterol. So what you have to know is that it's in a form of fluid. It's dynamic and flexible. It's not rigid. It's dynamic and flexible, and it adapts to shapes. It's flexible, like that kind of, it's dynamic. Its flexibility allows it to do a whole lot of things. For instance, the red blood cell, for instance, its flexibility allows it to maneuver its way through micro vessels, etc., to pass its way through. See how flexible it is, like this kind of smooth kind of movement. That is basically what the fluid mosaic model is. So let's do a very quick revision of what we've just done, a very quick brief summary of what we've just done. Okay, so we started by talking about the plasma membrane. We said the plasma membrane is made up of a phospholipid bilayer. The phospholipid bilayer has a hydrophilic head and hydrophobic tail. Then we said the main function of the plasma membrane is to serve as a selective barrier between the intracellular compartment and the extracellular compartment. So we talked about the various components of the plasma membrane. We said we have the peripheral proteins, the cholesterol, the glycoproteins, the phospholipid bilayers, and the transmembrane proteins. We also have peripheral proteins as well. Then we started talking about the components. So we talked about the phospholipid. We said we have the plasma membrane is divided into the inner membrane folding and the outer membrane folding. The inner membrane folding consists of phospholipids such as phosphatidylethanolamine, phosphatidylserine, phosphatidylinositol, and the outer membrane consists of phosphatidylcholine, and then sphingomyelin. And we talked about the functions. Then we came to proteins. We said proteins, we have the integral proteins, the peripheral proteins, and the lipid anchored protein. So basically it's the integral proteins and the peripheral proteins that are predominant. So the integral proteins, they are embedded within the plasma. In examples, we have the channel proteins, carrier proteins, receptor proteins, cell adhesion molecules, and enzymatic protein. Then the peripheral proteins, we talked about the cytoskeletal proteins. That is where we talked about the spectrin, the actin, the anchoring, and the protein bands. Then we have signaling proteins as well. Then we further move to talk about the glycocalyx. We talked about the antigenic property of glycocalyx. We said the glycocalyx consists of glycoproteins, glycolipids, proteoglycans, and polysaccharides. Then we talked about the fluid mosaic model, and finally we talked about the general function of the plasma membrane. Alright, thank you very much for joining me in this session. We shall discuss more in future videos. See you. Bye.